Assalam and a very good evening to everyone. I hope you have enjoyed yesterday's session. I am Sabah Sheikh, Assistant Professor at School of Pharmacy. On the behalf of Anjuman-I-Islam Kalseka Technical Campus, New Panvel, extend my warm welcome to all dignitaries, resource person, faculty members, participants from different colleges across the India to day 3 of 1 week AICTE-ISTE sponsored refresher program on multidisciplinary and translational drug development. To start with, as a tradition of Anjuman-I-Islam, I request Professor Abu Sufyan to recite and translate few verses from Holy Quran.
Okay, thank you, Professor Sabah. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم إن الذين يتلون كتاب الله وأقاموا الصلاة وأنفقوا مما رزقناهم سرا وألانية يرجون تجارة لن تبورا. The translation of these verses from Holy Quran is, verily, those who recite the book of Allah, that is Quran, and perform the prayers and spend in charity out of what we have provided for them, secretly and openly, they hope for a sure trade again that will never perish. Sadaqallahul Azim.
Thank you, Professor Abu Sufyan. These verses of Holy Quran are worth implementing in our life. Yesterday, we had a very interacting and engaging session on computer aided drug design, in which our resource person has discussed about traditional and modern drug discovery process. He also shared case study of work done in his lab on anti-cancer and anti-HIV drug development. Going ahead, today we have a very interesting session on introduction to biomarkers and drug development from bench to bedside. For this session, we have with us eminent resource person Dr. Mansi Pimple, and it's my privilege to introduce Dr. Mansi. Dr. Mansi is working as a field application scientist in Bio Nanogenomic, Florida, USA. She is molecular biologist with PhD in molecular medicine and pharmacogenomics from Nova Southeastern University College of Pharmacy, USA. She is awarded with Quarterly Excellence Common Value Award, Canon Primary Care Cancer Research Grant, NSU PAN SGA Professional Development Grant, President Faculty Research and Development Grant. She is actively involved in cutting edge technology in cancer research. With this brief introduction, I extend my warm welcome to Dr. Mansi and request her to start with the session. Dr. Mansi, it is over to you, ma'am.
Thank you, Sabah ma'am. Can everybody hear me well?
Yes, ma'am.
Awesome. I'm going to try to share my screen. All right. Good evening to everyone attending this workshop. It is my utmost pleasure to present this brief talk in front of you. Thank you, Dr. Sabah, for the introduction. I'm always grateful to Dr. Syed and Dr. Abu Sufyan for always having me and being welcoming. Ready to hear whatever thoughts I have to share about my experience in cancer research, as well as in the technology and cancer research field that I am in right now. So before we begin, I want to make sure that we keep this session interactive, and I have feedback coming from all of you. So if everyone can use their chat box, I want you to share with me as to your background. So do we have all teachers here, or if we have researchers as well? So if we have people who actively do research among the faculty today, just type in research and hit enter. So I just want to see what you all do. And if you don't do research, if you teach, that's fine. Just write teach and just hit enter. All right, we got one researcher. Any more? Okay, more researchers. This is exciting. All right, 4 researchers so far. We'll give everybody 30 seconds more. Okay, we just got academics. That's great as well. We got the mix as well, academicians as well as researchers. That's amazing. Awesome. So I'm so glad everybody's responding. Thank you everyone for your responses. It's really nice to see a mix of both teaching faculty as well as research faculty and faculty that do both. It's the reason I asked this, I wanted to make sure that whatever I talk about today is going to be relevant to everyone. You, even though you're only a teaching faculty, it's always nice to keep abreast of the latest technology that's out there that's helping biomarker research, and whatever experience I have in terms of the exploratory biomarker research that I've done during my PhD. So I will I'm just hoping everyone can see my presentation screen here. You
Yes,
already
we can
have.
see.
Thank you, Dr. Abu Sufyan. So you already have my introduction, so I won't go into it. I will straight away delve into our topic. So, biomarkers. I definitely don't want to insult all of your intelligence, because I know I have some really bright faculty and teachers from all over India. So I didn't want to put up the definition for a biomarker, but it's up there right now just to kind of give us a head start. We all know what biomarkers are, especially if you are a researcher. um, Sometimes finding a biomarker for the area of your interest is your sole aim. At least that's what was for us when I was a PhD student working towards my dissertation. So basically, the way we can define a biomarker is that it's basically a measurable entity, anything that can be quantified or can be detected. So it's either the amount of the presence of a particular characteristic or a particular measurable entity, or the presence or the absence of a particular entity is what we call a biomarker. Now, it can be indicative of a normal biological process. It can be indicative of a disease state. It can be indicative of a response to an intervention that you give, for example, a drug treatment. And it can also be indicative of a lot more things, like, say, toxicity or safety concerns. So biomarkers are kind of a window into the really complex bodily functions that are taking place in our body. So I have some pop quiz questions for you before we start off, just to see how our faculty participants think about biomarkers. So Dr. Said or Dr. Abu Sufyan, do you want to publish the first question? All right. So I want you to choose which of these options is not an application area of a biomarker. You have 4 choices. All right. We have some responses coming in. We'll give it another 20 seconds. People, you don't have to be right. Just give your opinion, and we'll talk about all these anyway in our presentation. So feel free to give your responses. All right. I think a lot of you have voted already. So that is right. A majority of you are right. Hemostatic is not an application area. So if you compare the choices that I gave, a biomarker can be a diagnostic biomarker, it can be a prognostic biomarker, or it can be a predictive biomarker. But hemostatic biomarker just doesn't, it's not a characteristic of what a biomarker can be. However, a hemostatic marker can definitely exist. That's a different question. So thanks for participating in the poll. It's really exciting that you probably know more on the area of biomarkers than I do, but hopefully I can share some new content as well. All right, so let's go ahead and talk about the types we just had our pop quiz on. So basically, biomarkers it's a really huge umbrella term which can be divided into certain sections to kind of understand what their actual function is. But bear in mind that they always overlap in terms of the type of biomarker that they are. So starting with diagnostic biomarkers, you're very well aware of, diagnosis of a disease being a very important aspect of giving the optimum treatment to patients and making sure we increase their quality of life and survival. Therefore, diagnostic biomarkers, I would say a lot of research is geared towards making sure that we make diagnosis of really complex diseases easy and readily available to patients. So a really good example of a diagnostic biomarker is gene expression profiling that is routinely used in breast cancer to detect type of breast cancer that a woman has, and therefore be able to treat it in a better way. In similar sense, your blood pressure is also a diagnostic marker, wherein a high blood pressure signals you have hypertension. So similarly, you have other biomarkers where they kind of they predict the risk of you getting a particular disease or a syndrome. So for a great example, and you probably might be aware of this, is your BRCA1 and 2 gene mutations that make you susceptible to breast cancer. So you'll notice that a lot of examples that I give are breast cancer related because that was what my dissertation work was based on. So I know examples related to those best, but there are tons of examples, and there are literally fields of studies on biomarkers for every disease type or every, say, metabolic pathway or genomics field has its own set of biomarkers as well. So a vast field, but I'm just trying to give the examples that are readily available to me. So prognostic biomarkers, another thing. So you are trying to predict the prognosis of a patient. How long is the patient supposed to survive? How long can a patient be disease-free? How long can they be in remission? Those are really important biomarkers for long-term or chronic diseases like cancer or other chronic diseases that you can think of. So predictive biomarkers, again, really important. Prediction is a way to kind of assess the risk and the treatment options for a patient well in advance. So if the person has a specific biomarker, you're predicting them to be at either a higher or a lower risk for a particular disease, or you are predicting that a drug will work better or will not work in a specific patient having that biomarker. A great example is, again, the BRCA1 and 2 gene mutation. The presence of those imparts sensitivity to ionizing radiation to patients. So by that, we mean breast cancer patients having that kind of mutation, when treated with ionizing radiation, will have a lot of, say, toxicity effects. So you can basically cater therapies based on such predictive biomarkers. Now, response biomarkers are nothing but the biomarkers that are tested as a response to a particular intervention, or they're tested as a kind of an endpoint to a particular intervention. A great example is your H1B/AC levels for diabetes monitoring, or your blood pressure, or even viral load for monitoring of antiretroviral therapies and their effectiveness. So the decrease in viral load, excuse me, is going to be able to signal that your antiretroviral therapy is working well. Then comes your monitoring and safety biomarkers, wherein monitoring biomarkers will let you again monitor chronic diseases and make sure they're in check or patients remain in remission. And safety biomarkers, as the name indicates as well, in itself, they are an indicator of toxicity in patients or certain risk factors for patients, either due to an intervention or due to their disease state. Great examples: bilirubin, serum creatinine, or urine and kidney biomarkers are always toxicity indicators, because as you'll notice, toxicity is usually linked to your drug being metabolized in your body. So that's a very broad categorization of biomarkers, but I really like this diagram that I found on a biomarker company's website, where they have done a great job of kind of putting biomarkers in context. So if you're looking at the slide, the top panel tells you the path from taking a drug moiety from a laboratory bench to the patient bedside. So there's, as you know, a huge process involved, which is also a multi-billion-dollar process from taking that product from the bench to the patient. So in these, there are a variety of steps involved. As you know, there's basic research involved. I am sorry, I'm going to close the poll. I don't know why it's here. All right. Sorry about that. There's basic research involved where you actually test the efficacy of the biomarker, or you test the efficacy of the drug. So for example, and then you need markers that give you an indication of how effective the drug is, if the drug is causing any toxicity, if the drug is actually helping in changing the disease marker, which is, say, for example, either reducing the tumor burden or reducing viral load in this case. So you can see biomarkers here are important in every single step of the way, from basic research to clinical trials to actually even after you administer the drug to the patient, and once it's in the market. So safety monitoring continues constantly throughout the process. We want to make sure the drugs we give to our patients is safe. In certain chronic diseases, prognostic markers are always evaluated. For example, in cancer, if you are trying to evaluate the efficacy of a drug.
What happens next with the vaccine? Safe to say that the...
All right, I think we had some background, but that's gone now. Thank you for muting. So so I was saying in cancer, a good indication of prognosis is survival time. So whenever a drug is tested for efficacy in treatment of cancer, survival time is always measured, as you know. So that's kind of a prognostic marker as to how much a drug can prolong survival time. So basically, biomarkers, and this is all in a example of a clinical setting. But as we progress throughout my presentation, I'll be talking more about just biomarkers in the exploratory research phase. So we'll see about that. All right, it's time for our pop quiz question number 2. This is just to get a basic idea. I think there you go. Question number 2. So feel free to give your examples really quick. If you had been listening to my presentation, you will know the correct answer. And if you don't, it's completely fine. We will see what the right answer is. So go ahead and cast your poll. All right, we have a few responses in.
Early committee had realized that all these...
All right, a few more seconds, everybody. Please cast your responses. It doesn't have to be correct.
you can see the poll out there, right, on the screen? Like, I don't see anyone responding yet. You can see it,
I
right?
can see the poll. Yeah, I can see the poll, Dr. Sai.
Okay.
Are you? Oh, I can actually share my screen. So if everyone wants to see what's going on, but all right. I think we've given it a minute and a half, and that's pretty good. So interesting results for the poll. I see that most of you have chosen either diagnostic or a response biomarker or a monitoring biomarker, which actually are all correct. So the correct answer is E, all of the above. As I said, if you were paying attention, biomarker types are usually overlap, because what is a diagnostic biomarker can also continue to be a monitoring and a response biomarker. So in this case, viral load is definitely a diagnostic biomarker where you can test the viral load and see if the person has an infection. You can monitor the viral load, which is especially important for patients like HIV patients, where antiretroviral therapy's effectiveness is monitored. And again, the response to antiretroviral therapy is monitored with the help of viral load as well. So in this case, all the three answers are correct, which it's a diagnostic biomarker, it's a response biomarker, as well as a monitoring biomarker. So that was exciting. Thank you for participating, everyone. On to our next portion. So I'll try to be relatively quick in the interest of time. Basically, you all are aware of the central dogma of research of biology, which means the entirety of life centers around basically the conversion of nucleic acid in the form of DNA in most organisms, which is transcribed into RNA, and RNA are ultimately translated into protein. These proteins are nothing but effector molecules that go ahead and actually carry out the job that they were made for. So you can think of DNA as being the script, RNA as kind of being the intermediary people, and protein being the actual actors who carry out the job. Now, basically, biomarker discovery or biomarker research, I could think of, is centered across three major areas in basic biology research, which is genomics, proteomics, and metabolomics. So I'm sure there are more areas, and people love to have omic names for all the fields out there, which is super interesting. However, today's talk, my focus has always been on genomics. And I have been focused in my research to find genomic biomarkers for breast cancer, as well as the experience I've had after my research, as well, that also has been focused on kind of manipulating genomics or trying to uncover the secret in genomics by using cutting edge technologies to actually discover biomarkers that can either be used for diagnostic purposes or can be used for drug development. So this is where my expertise lies. So after I finished my dissertation research on breast cancer, I ended up using next generation sequencing. Sorry about that. I ended up using next generation sequencing a lot in my research, where I used RNA sequencing to study the actual DNA repair pathways, and we'll go more into this later. But next generation sequencing has emerged as the giant in the biomarker discovery field, or just in the cancer research field. Or, not only cancer, it's now being applied to every single research area in the biology field. So I wanted to give you a brief idea of what technologies you have at your disposal out there that you can use to further your research. The NGS world is, to put it very briefly or naively, is basically split into short read sequencing and long read sequencing, which means the way next generation sequencing works is that it was basically inherited from Sanger sequencing, where you could sequence a specific fragment of a specific length at a given time, but the process was slower. It was in its infancy, but just the fact that we could uncover the sequence of DNA was itself in itself a huge discovery. So after Sanger sequencing, we have some really new, exciting, and fast-paced sequencing technologies now available in the market. And the best example of that is Illumina sequencer. So Illumina is a short-read sequencing technology, as you're probably aware already. What that happens is it can read DNA in short fragments of up to 300 base pairs. The capacity of every machine offered by Illumina differs, and the throughput differs. But basically, all you can get is the maximum read length of about 600 base pairs if you read them both forward and backward. So paired-ended sequencing is more accepted than single-ended sequencing, just because you get the validation of having two reads read the same region, which means higher accuracy in reading that particular segment of DNA. Now, with the help of short-read sequencing, getting millions of reads in a single run, you're able to do a lot downstream. So you're able to do a lot also with the starting material. So you can sequence DNA. You are now able to sequence RNA. You're now also able to sequence small RNA molecules, which have been a more recent discovery. And we'll talk about microRNAs later in my presentation, but Illumina sequencer is capable of basically sequencing any fragment of DNA, but its major limitation is it'll sequence it just up to a short length. So its read length, which is the actual term used for the length of the fragment that can be sequenced, is short for Illumina, and that's where the limitations come in for Illumina. Although it's a wonderful technology, widely applicable, you can use it to sequence fragments that have been generated from complex assays involving proteins like ChIP-seq, ATAC-seq. So ChIP-seq gives you a good idea of transcription factor binding process. So the place where a protein binds to DNA, that can be detected with the help of a ChIP-seq assay, where the protein interaction which is followed by sequencing. ATAC-seq is another really cool assay where you can actually determine what proportion and what segments of the DNA are in an open fashion. Because if you all know, DNA is usually present in a bound fashion. It's bound to nucleosomes in the form of chromatin. This needs to open up in order for DNA to express itself, to make RNA, and then to make proteins. So getting an idea of an open configuration is a great research tool that you can use to actually study what prompts DNA to open and close, what are the processes involved, what happens when that actually needs to happen in the body to express RNA and proteins. So anything that is downstream, any fragments of DNA or RNA generated can be sequenced on Illumina's machine, although they're short reads. This led to the advent of long read sequencing, whereby PacBio, Oxford Nanopore, and technologies like that, they kind of found out ways to sequence really, really long fragments of DNA. So with PacBio, an average read length, or the length of DNA that you can sequence at a given time, reach up to about 20,000 kilobase pairs compared to the 300 or 600 that I spoke about with Illumina. So in one read, you're able to sequence about 20,000 base pairs on average. And the longest read with PacBio can reach up to 100,000 or 1 lakh base pairs for understanding purposes, which is pretty astounding if you think about a jump from 300 base pairs to 1,000 base, 1 lakh base pairs. So from there, Oxford Nanopore has similar technologies, but Oxford Nanopore sequencers, if you see here in the little diagram, and I'm sorry for the small pictures, but I didn't want too many slides. So the Minion by Oxford Nanopore is a sequencer that you can literally plug to your laptop. You can set it on the bench next to your laptop, load your sample, and the results will populate to your laptop. I think that's marvelous, and that's really mind-blowing as to where technology has reached now. So that is long read sequencing for you, and then there's more exciting sequencing applications where you can all these technologies that I spoke about give you an average idea of the genome or the transcriptome. What if your expression or your genome varies by cell type? So say, for example, we have always known that certain phenomena are tissue specific. The different tissues or different organs in a human body have different functions, thereby have different expression of proteins, have different expression of nucleic acid. So if tissue specificity is a huge factor in determining, say, for example, what biomarker might be important in, say, colon cancer versus pancreatic cancer, just for the sake of an example, you need to study the specific mechanisms that happen that are specific to either the pancreatic tissue or the colon tissue. So in that case, that's where your single cell sequencing comes into place, where 10x Genomics has found actually it has come down from an older technique called DropSeq that was brought about by Bio-Rad, if I remember correctly. But basically, you can sequence nucleic acid, either DNA or RNA, at a single cell resolution. Each cell is kind of uniquely barcoded with the help of a UMI, that's what they call it, unique molecular indicator. After barcoding each cell and then sequencing all the fragments of nucleic acid in all the cells, you are then able to bioinformatically parse that information out to be able to tell what fragment of a particular gene or an RNA originated from what particular cell type. So basically, it'll give you a cluster of cell types, and I wish I had an example to show you, but it'll give you a cluster as to what kind of cell types you have, how many cell types you have, and what is the expression of your particular genes of interest in those cell types. So I think that is pretty cool. And then nanostring technologies takes it to a next level where sometimes you want to study transcription or gene expression in the context of in a spatial context, which means you want to study expression in terms of the position of that particular piece of tissue. So if you can see in this figure right here, digital spatial profiling actually combines immunohistochemistry along with sequencing, or gene expression profiling, whereby you can study the different tissue types and molecular expression of certain biomarkers, and at the same time know what their expression is relative to other areas of that same biomarker in the same tissue piece. This is one of the coolest technologies that I personally, have known about, because this is exactly what my research was based on, and I wish I had this back then. But whatever the field is coming up with is really exciting, and it is really going to go a long way in helping researchers uncover the deeper secrets of, say, for example, the human body or certain diseases that have been elusive. So moving on, and I'll try to be a bit quicker, cytogenetic assays are another huge section, and that's where my current work lies in. It's in this area of cytogenetic assays. So basically, you've known now with next generation sequencing, you can look at certain, albeit very small fragments. Now DNA is huge, right? It's comprised of billions and billions of bases, and you with NGS, it has a limited capacity to study the entire genome on its own. Cytogenetic assays have been traditionally used to study the genome on a chromosome level or on an entire whole genome level. So starting with the chip-based arrays is what is very commonly used as an alternative to NGS, because A, they are cheaper, B, they're quicker, and C, they're more easily, say, for example, they're easier to run than the NGS assays. But however, the biggest consideration in running chip-based assays compared to NGS is cost. They're much cheaper than NGS, although NGS is now at a point where it is so readily available that its cost is dropping to almost like $100 per genome, which was unimaginable at least like 5 to 10 years back, where the genome was quite expensive. So coming back to chip-based assays, chip-based assays are a great way to quickly interrogate the entire genome, because what they do is they have probes specific to certain regions of interest that are present on the DNA, or certain RNA molecules that you are interested in studying. So you can have chip-based assays specific to a disease type. You can have chip-based assays specific to a metabolic pathway. There's a lot of things that you can do with these chip-based assays. However, they are again limited by their ability to interrogate the entire genome, because you will only have those many areas represented in the chip as many there are probes on the chip. So there's that limitation, and then karyotyping and CGH arrays kind of overcome that limitation. However, their resolution or their resolution is not that deep as SNP arrays. So SNP arrays and chip arrays, or they can go up to a single base pair resolution, whereas karyotyping and CGH or FISH give you an idea of the chromosomal level. So you all are aware of karyotyping. Basically, it's a way of detecting chromosomal abnormality. So there are certain banding patterns that chromosomes have, which trained cytogeneticists are able to identify after arresting the cells in metaphase and then staining the cells for the appearance of those bands, and then looking at them under a microscope. So a trained eye can identify what a normal chromosome and banding pattern looks like versus a diseased banding pattern. So although this requires a lot of time investment and training, FISH is just an adaptation of karyotyping, along with so it's basically targeted karyotyping, wherein you specifically target fluorescence probes to a specific area of interest or locus of your interest. Say, for example, you are studying leukemia, and you want to look at the BCR-ABL fusion, and you will specifically generate a probe that goes and finds that region, and then you can see it under a fluorescence microscope just when you have the chromosomal smear like this. So this is very powerful if you just want to interrogate a few regions that you know of in a sample. However, again, it fails when it comes to say overall interrogation of the genome. This is where bio-nanogenomics comes in, and I was super interested in working for them, and I'm so glad that I do, because their technology is actually really mind-blowing. If you has anyone ever seen a DNA fragment of DNA by themselves? I don't think so, because I'm sure no one has seen a fragment of DNA by themselves. However, in bio-nanogenomics technology, if you see each of these vertical lines that are blue in color with those green dots, it's a single DNA molecule. So what bio-nano's technology does is it kind of adds fluorophores, which occur repeatedly throughout the genome, these green little dots that you see, and then the backbone of the DNA is counterstained to be able to detect those green labels. This kind of, the DNA after labeling is then passed through really small nanochannels and is linearized, by which you will see these really long stretches of molecules. This is when this is, after imaging these really long stretch of molecules, they can be converted into bioinformatic data or de novo assembly maps that can, give you a whole genome overview. And the best thing about it is, remember when I told you the longest read you can get with the PacBio machine is about 100,000? Even that's like the longest read. It'll just be 1% of the total reads. Average read length is 10,000 base pairs. With BioNano, BioNano only analyzes DNA molecules that are more than 150,000 base pairs in length, which means 150,000 base pairs and above. The longest molecules that we know can be isolated is approximately 2 megabase pairs, which is huge. It's about 2 times 10 to the power 9, I want to say, but they're really long molecules, and this is really the power of the technology. And long molecules enable you to phase certain genomic changes, which means phasing is an idea where certain genetic traits are inherited together, and the longer the molecules you have, the better the idea you get as to what changes or what segments of DNA lie adjacent to each other, and the validity of those maps that are generated also increases with the longer reads you have. So this is where technology is taking us right now, and this is kind of the end of my first part of introduction to biomarkers. I know I could have gone on and on, but in the interest of time, I would like to open the floor for any questions on this part of my presentation.
So the faculty members can either type in their questions or they can unmute themselves. Either way, it has been a very interactive sessions where Dr. Mansi has given some very nice insight about the current work in the biomarkers field, essentially. So Dr. Mansi, essentially, so some of the technologies that you talked about, so are these, so where is in terms of the current state of they being used for, as for patient selection criteria? Are they still more into more of a research investigative mode, or more into using it in the clinical trials for enriching patient population, or being that as a predictive marker? And where are we with this use of these current technologies that we just spoke about?
Sure. Yeah. That's a great question, Dr. Said. So the ultimate goal of developing all these technologies is being able to finally take them to the clinic. However, as you see, these techniques need a lot of technology investment, a lot of personnel investment, a lot of, say, for example, resource investment. So it is at this point a bit difficult for every clinical lab that exists around the globe to have such expertise, such such machine availability and the knowledge to operate them, and also the resources to operate them. So as of now, I want to say the access to obtaining such technologies and their benefit for advancing research has increased dramatically. So my previous position where I used to work, it was a service provider, which means these service providers exist across the globe. They usually have a laboratory with tons of such machines, and they provide these sequencing services to customers for a fee. So basically, people across the globe can send samples to such a service provider company and get sequencing results just basically sent to them informatically, or just via cloud transfer. So it's become really easy to access them. However, their use in the clinic, I think, is still going to take some time. They have recently started using NGS in clinical labs, and even BioNano, my company that I work for, we have started putting our footsteps in the clinical area, where we have a few machines in clinical labs that do diagnostic testing every day. But it's a work in progress that's going to take some time, but we are definitely at the point where we have started it.
Great, thanks. Dr. Nikat had a question in terms of the roles of biomarker as such in, more into diabetic radiotherapy. I don't know what she meant about it. I mean, I think diabetes, there are tons of biomarkers that have been used. Dr. Nikat, can you elaborate on that in terms of diabetic radiotherapy as such? Would you like to comment on it, Dr. Mansi? I mean,
yeah. So I wanted to say that I'm definitely not an expert in diabetic radiotherapy or diabetes at all. However, the role of biomarkers cannot be, emphasized enough in any field, literally. So whatever field you choose, people will be in the lookout for biomarkers to make clinical treatment more effective, or probably more readily available, or diagnostics more easy for clinicians to be able to diagnose patients in a timely manner, to make sure they're treated in a timely manner, and that they have a good quality of life and survival. So I'm not sure if I can answer a question in terms of like the field of diabetes or diabetic radiotherapy, but let me know if you need me to elaborate on that, Dr. Nikat.
Yes, madam, it's typing mistake, diabetic retrotherapy.
Oh, I'm actually not even sure what diabetic retrotherapy is, because I'm definitely not an expert in diabetic retrotherapy. So I can get back to you on that, but my role is basically focused on biomarkers in terms of cancer research, and that's where I'm going to cater the second part of the talk.
Okay.
Great. I think there are no further questions. I think we can move ahead, Mansi, to the next session.
All right, sounds good. So when I talk about biomarkers and drug development, as I said, my talk is mostly going to be focused on cancer research, because that's the area I'm in right now, and that's where I've been from the time I started my research. So to give you all a topic to give you all an idea as to why I keep talking about breast cancer and cancer research, this was my dissertation topic. So when I did my PhD research, I studied sporadic breast cancer cases, which means breast cancer cases that cannot be attributed to a specific etiology or cause. So breast cancer, we know, have multiple types. They're usually characterized by expression of a specific biomarker in the form of hormone receptor. So they're either estrogen receptor positive, they're either progesterone receptor positive, or a combination of those two, or they are HER2 receptor positive or negative, or a combination of all of these three. And then there are different other biomarkers too that are used to classify breast cancers. But what about the cases that show none of these classifications, or rather, more importantly, sporadic cases means that cases that don't have a familial history of breast cancer. So in patients where you have an inherited gene that tells you that you have a risk factor for breast cancer, or you have a history in your family that people had cancer, and that's why you are at a higher risk of getting cancer. But what about people who get cancer with no familial history, no hereditary genes in them that could explain why they got the disease? Such diseases are known as sporadic. So basically, our goal was to try to pinpoint a biomarker that'll help us diagnose sporadic breast cancer cases, treat sporadic breast cancer cases, and prevent the metastasis of sporadic breast cancer cases. So our lab focused on nucleotide excision repair, which is one of the 5 major DNA repair pathways. DNA repair plays a huge role in cancer, and we will see that in a moment in my slides further. So we already knew that nucleotide excision repair was impaired or was reduced in early stage breast cancer cases from the studies that our lab had done previously. I now wanted to focus on seeing if I can track what is happening when a cell converts from a normal cell to a tumor cell. So can we pinpoint a specific biomarker or a specific gene or a pathway or something that changes and can tell us that a particular tissue has become tumorigenic? Now, why do we need to show that, right? You usually, clinicians have a pretty good handle on diagnosis, and women get mammograms to make sure they don't have breast cancer. And if they do, they will show up in the mammograms that you have regularly on a yearly basis. So why do we need this research? Well, turns out that the way cancer works is really interesting. So there are theories out in the cancer field where cancer is called as, there's a field carcinogenesis theory, or mosaicism theory, which is still there. Mosaicism is still a fact found in every tissue type, wherein basically there's a field effect where a cell that goes past the tipping point of being converted to a normal from a normal to a tumor cell, we can only detect it when it is morphologically, or it is evident, and a pathologist can find certain changes that a cell goes through after it's way past that point and has become a tumor. We know that with research, researchers have found that cells already go through a lot of changes at the genetic level, which make them abnormal and more susceptible to becoming tumorigenic, and then us being able to actually identify those tumors and then treat them. By the time they've become a tumor of a specific size, it's kind of too late, because this model kind of explains what is happening. So this is a very simplistic model that our lab came up with, and explains my research in one slide if I had to do that, which is usually very difficult. But say, for example, that the first row here is just a tissue that a normal breast tissue that has one cell that has acquired one mutation. So cancer has been known to be a phenomenon that occurs due to accumulation of numerous mutations, which are usually driver changes and passenger changes, which means there's one change that drives, it's the tipping point from normal to tumor. And then because of that change, it gains more mutations, making the cell unstable or the tissue unstable, and therefore making it into a tumor and allowing it to grow uncontrolled. So if we imagine that first change, the tipping point, happened in this green cell here among that normal tissue, that green cell is also proliferating, but at this point, we're not able to diagnose it. That change is basically happening at the genomic level, or at the level of your nucleic acids, or say proteins that are made. So they aren't still morphologically tumor that a pathologist can identify being abnormal. So that little green cell there divides. Among that, there's one cell among the green cell that gets the second mutation. That divides. You now have two populations of abnormal cells. There's then another cell among that has a third mutation. There are now three or more populations, and bear in mind that each of these cells are still growing and dividing. So if you put that in the context of the breast tissue, you have a field of abnormal cells at the genetic level that finally are going to get that final driver mutation that's going to make it into a tumor phenotype, which means a pathologist is going to be able to identify if he samples cells from this blue section of the tumor. If he samples the other cells, they're going to look normal to the eye. However, there are genetic abnormalities that lie between those cells that make them abnormal. This tissue is usually left out in the breast when you have tumor removal surgery, because usually surgeons or pathologists can't find any reason to remove normal tissue because it looks normal. However, surgeons usually also cut out a 2-centimeter margin along with the tumor that they cut out when they do the surgery to remove the tumor. That actually takes out a lot of these normal cells, but not abnormal cells at the genetic level, but not all of them. So what if we had a predictive biomarker that could tell us when a cell is becoming abnormal and is not functioning normally and is on the path to becoming cancer? Wouldn't that be wonderful? We could then just prevent cancer in that way. It's a very far-fetched scheme, but that was our entire goal, to see as to if there is a specific phenomenon that's happening which can predict why or when that particular tissue is going to become tumor, and if we can specifically target drugs to stop that. So you can see that what's happening with biomarkers now, more than going into, it's still there with diagnostics and other application areas that I mentioned, but it is more being used for personalized medicine and precision medicine. So for example, basically, you you cannot treat every patient with an empirical therapy that is meant to treat every single breast cancer patient that exists on this planet. So not every cancer is the same, not every person is the same. So there is no explanation for us to give them the same treatment, right? So you have to cater the treatment based on the type of cancer they have, based on the individual characteristic that a person has, based on, say, their ethnicities, their racial origins, because even that dictates a lot of genomic expression of of a lot of proteins and pathways that are different between certain racial ethnicities. For example, Ashkenazi Jews are more prone to getting breast cancer than any other ethnicity type. African American women are definitely more susceptible to getting breast cancer than other ethnicities. So there are specific characteristics each individual has which need specific treatment. This is why we are now moving towards precision medicine, and biomarkers are a great way to do that. And they are kind of emerging to be the only way to do that. So this chart here basically just gives an example of the same breakdown that I had for biomarker types previously in the first half, where you're still using biomarkers to assess risk, to diagnose, to predict prognosis, to do some predictions and do monitoring in breast cancer. However, you are now catering each section to a specific set of patients. So for example, if you look at prediction as to how a drug is supposed to respond in a patient, you know that a patient that expresses the HER2 receptor is going to benefit more from a HER2 receptor antagonist like Herceptin than other patients with breast cancer that don't express the HER2. So that's why discovery of the HER2 biomarker was a great breakthrough in breast cancer research. Similarly, there are breast cancer patients that express estrogen receptors in great amounts. So in that case, it's estrogen receptor antagonists like tamoxifen is a boon to those patients, and it's called targeted therapy. Basically, you're targeting a specific receptor, and you're targeting a specific population of breast cancer patients that have the expression of that particular receptor. So biomarkers everywhere, wherever you look, there are biomarkers. Monitoring and prediction is also a huge area where biomarkers are playing a big role in breast cancer. So for example, if you look at these two variables right here, the one on the monitoring, they are basically nothing but certain cancer specific antigens that are found in the bodies of breast cancer patients that these tests can basically screen for, and we can monitor the remission of cancer patients. So say, for example, a patient went through chemotherapy, radiation, and is now in remission. We need to make sure they stay in remission and do not go into a relapse. So these kind of monitoring tests with specific carcinogenic antigens and their levels in the bodies of patients who have been treated are a great way to kind of keep making sure that they stay in remission, have longer survivals and a good quality of life. And if at all they see the presence of the disease coming back, it can be treated in a timely manner and not left until it's too late to kind of save the patient. So another great way of predicting as to how aggressive the disease will be, and these are really popular in the breast cancer world, there are certain diagnostic or gene expression assays that kind of predict how aggressive your cancer is going to be, how likely is it for your cancer to reoccur, and how bad is it going to be overall. So certain assays like Oncotype DX, MammaPrint, these are widely used in the breast cancer field to assess aggressiveness and to assess the likelihood of recurrence. Same way, risk assessment, genetic typing will give you an idea if a breast cancer woman or her family members have the BRCA1 or 2 gene, which will kind of help her kind of prepare for the possibility that the person is at risk for getting breast cancer if they don't have that at the moment. So you might have heard of Angelina Jolie. I don't know if you have heard or not. She's a very popular Hollywood actress. She discovered that she had this BRCA1 and 2 mutation, and she underwent a double mastectomy, complete removal of her breasts to make sure that she does not get breast cancer. I would say a very smart woman, because not everybody is really open to the idea of complete mastectomies. They are usually very invasive, and there's a psychological component to it as well. So, but you have the presence of such tests gives you more control as to what you can do to prevent or reduce the risk of you getting such deadly diseases that can severely affect your quality of life and the quality of life of your family. So some great examples here of what biomarkers are and how they help actually in a particular disease field. And here I took the example of breast cancer. So pop quiz time again. We have our question number 3, and I promise you this is the last question where I'll ask you all to chime in. If there we go, we have our question number 3. So which of these it cannot be called as a biomarker? So if you've been paying attention, you will probably remember that I talked about most of these. So feel free to chime in, doesn't have to be right. We'll discuss it anyway when we have responses in. All right, a couple of responses are in already. We'll give it 30 more seconds. So just for those who are not familiar with the acronyms, PSA is prostate specific antigen, HCG is human chorionic gonadotropin, the third one is tritiated thymidine, and the fourth one is HER2. Alrighty, quite a few responses are in. Another few seconds if someone wants to make a last minute vote. I see some last minute votes trickling in. Come on, faculty members, you can do this. All right, I see the responses are now slowing down. So let's see what we got here. Majority of you got it right. So tritiated thymidine is not a biomarker. It's basically a radiolabel, which can help you target a specific region, or it can help you in radioactivity assays, but it's not really a biomarker. So prostate-specific antigen is definitely a biomarker used for the diagnosis and monitoring of prostate cancer. Human chorionic gonadotropin, for those who chose that this is not a biomarker, it definitely is. Those over-the-counter pregnancy tests, they are basically used for diagnosis of whether a woman is pregnant or not, and they test for human chorionic gonadotropin, so it's definitely a biomarker. And HER2, we just spoke about HER2, so HER2 is definitely a biomarker too. So great job for those who answered it correctly. For those who didn't, no worries. That's why we're having this lecture. So let's move on to our next slide. Thank you for participating, everyone. Oops, sorry about that. All right. So kind of wrapping up as to why biomarkers are important in cancer. So you've probably seen a lot of you do cancer research too. So I see a lot of researchers here as well. Any cancer researchers here in among our researchers? You can use your chat box to interact. Let's have some cancer researchers, if at all, in our faculty. Anyone? Ah, no cancer researchers. That is very interesting. All righty. Well, all the more reason why we should, continue with this talk. All right. So, why is cancer such a huge pressing problem? People are like, why don't we have the cure for cancer already? Because cancer is a super smart and evolved disease. So it has really learned to evade all the natural checkpoints that our body has developed to make sure that nothing of the sort happens. And our body is pretty amazing in doing that too. So, for example, these are Hanahan and Weinberg. They were the kind of the pioneers for discovering the hallmarks of cancer, which means specific characteristics that enable cancer to do its destructive duties, which means keep growing uncontrollably, spread to other areas of the body, and ultimately reduce the functioning of organs and result in the death of an individual. So how does cancer do that? So basically our body has built in a lot of check and cross check mechanisms, and it is just amazing to see all these mechanisms kind of work in perfect synchrony to make sure that nothing goes haywire or nothing becomes out of the ordinary when it comes to cellular functions in the body. So what happens when a cancer cell is about to develop? So there are certain cells that there are certain signals in the body. When cells touch each other and in a tissue, there's cell to cell contact, and that kind of tells cells that, hey, we have enough cells now, you need to stop growing. But cancer cells kind of turn those signals off. They have an off button, and they don't hear those signals, and they are like, okay, I don't care if there's no room, I'm just going to keep growing. So they kind of evade the proliferating signals or the anti-proliferating signals that the tissue is giving them to not grow, and they keep dividing perpetually. So they also evade growth suppressors. So our body also has a lot of genes and proteins that kind of act as tumor suppressors. A big example is P53 gene. I'm sure you would have heard of P53 if you are a cancer researcher. Even if not, it's a very common tumor suppressor gene that is found to be mutated almost all of the times in cancers that are advanced stage. So fun fact, I know that I've heard that elephants have 50 copies or even more of P53 genes. So it's a point to wonder that do elephant get tumors? I don't know. I'm gonna have to check it out. But it's pretty amazing that these pathways are kind of conserved through species as well, and they're so robust in making sure that they protect the development of cancer. However, there are some smart and evolved cells that even evade such checkpoints. They activate certain mechanisms that cells are not supposed to do usually. So normal cells don't go about invading other tissues and then making that their home and then starting to grow there, or giving certain signals to kind of make new blood vessels for themselves and their partners that are growing unchecked. So that's a process called angiogenesis. There's another process where a very important one, apoptosis and necrosis, whereby any cell that has any deleterious change that is going on, or a mutation that, if survives, will cause trouble downstream or will be harmful to the body will try to kill it by either apoptosis or necrosis or using certain killer cells. So resisting cell death is another way a cancer cell survives. So a few more emerging hallmarks that are newer is, genome instability is a big one, is my favorite because that's what I've studied. So DNA repair pathways are the ones that make sure that our genome, the integrity of genome stays constant, that there are no mutations, and if there are, they are repeated in a timely manner so that they're not carried forward in future generations of cells. But cancer, as I mentioned, it is so smart that it has evolved to kind of turn that off as well. They can kind of survive with those mutations and still keep dividing What happens is it makes the genome unstable when it does that. So if you remember, I said that we found that DNA repair was low in early stage breast cancer. Well, you see now why. It's a major it's a major characteristic of how cancer develops and keeps growing. So there's tons of other ways that cancer kind of evades the normal checkpoints of the body. But this is where biomarkers come in. So all of these pathways have specific effectors that are now being targeted to develop specific therapies. So for example, taking the example of genomic instability and DNA repair, the fact that BRCA mutations in a breast cancer patient make the patient more susceptible to ionizing radiation usually happens because they have a deficiency of a specific DNA repair pathway. It is called double-strand break repair, and double-strand break repair usually happens when you either give a strong chemotherapy to DNA or to patients, or you give them ionizing radiation. So we now know that mutation in BRCA1 and 2 disables the double-strand break repair pathway, which makes the patient more susceptible to ionizing radiation. Now, what if you target that particular pathway and make cancer cells so weak that they cannot repair themselves and they die? That's exactly what PARP inhibitors do. So PARP inhibitors basically make the cells more sensitive to chemotherapy. They make the DNA the DNA to a point that it cannot repair itself and the cell cannot survive, thereby killing cancer cells. It also takes advantage of the fact that BRCA1 and 2 mutated cells cannot repair DNA and thereby cannot overcome the toxic insult that you give through chemotherapy. So PARP inhibitors are usually given in conjunction with other chemotherapeutic drugs in breast cancer, where you know that they are carriers of BRCA1 and 2 mutation. So kind of taking advantage of the already present mechanism of a particular disease state. So that's just one example. Every single hallmark that's mentioned here has a class of drug that has been developed by taking advantage of specific biomarkers in that particular pathway. The generating of blood vessels that I mentioned that cancer cells do, well, guess what? They have to use a specific molecule called VEGF. Now, VEGF signaling is very important for generation of new blood vessels, and this is blocked by VEGF inhibitors, which are now used as drugs of treatment for a lot of cancers like prostate. You can see that every single classification has a drug that has been developed by targeting a biomarker. So in short, can biomarkers be used towards drug development? Yes, they absolutely can. You saw so many examples just right now. To conclude, I will just take you through two very cutting edge technologies or basic science research advances that are now being used in drug development and as biomarkers. So one of them, and I spoke to you about this a bit earlier, are microRNAs. So what are microRNAs? They're nothing but small RNA molecules that are usually present in the normal human body. So if you know about the basic classification, we know that genetic mechanisms are basically conversion of DNA to RNA, and RNA DNA both comprise of our genetic material, and there are genetic ways of expression of these molecules that ultimately result into formation of protein. Now, there are certain mechanisms that are certain changes that are also called as epigenetic changes, which means they are certain heritable changes that themselves do not do any expression, but they regulate the expression of these DNA and RNA molecules. So miRNA is a great example of an epigenetic mechanism. Epigenetics is a huge field right now, and I had the chance to dabble in it a little bit. Methylation, so turning the genes off by methylating DNA, is another epigenetic mechanism. Histone deacetylation. So DNA is wound around histone proteins. The acetylation or deacetylation of histone proteins determines what DNA will be available for transcription and what will be closed off and will not be able to convert into RNA and then protein. So histone deacetylation is another epigenetic mechanism by which genes or DNA can be turned on and off. And these changes are usually inherited. So they're heritable changes that are passed on from cell to cell, generation to generation, and they are actually reversible. That's what makes them epigenetic. So a mutation in that gets carried forward in a cell is not reversible. It stays in the DNA. It gets carried forward and stays a mutation. Epigenetic changes are reversible. So an miRNA can perform its job and then can go away. That's the beauty of miRNA here. So not going to get into details as to how the miRNAs are made, but basically they have two functions, or they can be used in two ways. So miRNAs are nothing but small RNA molecules. They are single-stranded as opposed to double-stranded DNA. And what they do is they go and bind to the 3' UTR or even the 5' UTR untranslated region of an mRNA, a messenger RNA, so that it blocks the conversion of RNA to protein. So it blocks the translation of RNA, and therefore the protein will not be produced. So this can be actually taken advantage of in two ways. Say, for example, you have an oncogene, right? Which if expressed in a cell is going to make it tumorigenic, or is going to convert it into a tumor. Now, using an miRNA to block the expression of that oncogene, if it's being expressed in a cancer cell, is a great treatment modality to, or is a great way of kind of using miRNA as a biomarker where that oncogene is being expressed. On the other hand, if you have tumor suppressing genes that are being stopped, you can add miRNAs that will kind of enhance the effect. Or if there's an miRNA that has been suppressing a tumor suppressor gene, I know it's complicated, but try to imagine a strand of RNA that's supposed to be stopping tumor, an miRNA blocking that strand of RNA, which is supposed to be stopping the tumor, thereby the tumor is now growing. How about you make an antisense miRNA that binds to this miRNA and kind of releases the tumor suppressing messenger RNA to go and stop the tumor? So there's a lot of ways that this can be done. And a lot of companies are now taking advantage of these miRNAs to actually target specific specific disease types, and they are now in clinical trials. So this is a pretty recent list from 2019 from a paper that I've listed here. Quite a few companies are now using specific miRNAs that are found in the body. And the beautiful thing is now you have miRNA databases where you can actually go and look, try to find the miRNAs that have already been discovered. There are also novel microRNAs that have never been discovered, and that's where your microRNA sequencing comes into place. So, that's a great way to keep discovering novel biomarker miRNAs to be able to kind of see if they would be better treatment modalities for certain cancer types or certain difficult to treat disease types. Another really cool example, and this is my last example for today, CRISPR-Cas9. I'm sure everyone here must have heard of CRISPR-Cas9. The two lead scientists that discovered CRISPR recently got the Nobel Prize. Basically, CRISPR is nothing but a gene editing mechanism. We know that 3 patients in the US were successfully treated with edited cells using the CRISPR-Cas9 technique. We were able to prolong their lives for a few months, so which shows that this gene editing technique definitely has promise, but it has its own limitations. You have to be able to exactly target that particular area of the gene segment that you want to edit. Now, CRISPR-Cas9 has also known to have what you call as an off-target effect, which means it can bind to regions that have similarity with your regions of interest, and that can be anywhere in the genome. So say, for example, it binds to regions of interest and goes and binds to something else that is a super important gene necessary for your body, you are now kind of creating more trouble than you are trying to, solve. So CRISPR is a huge field. There's a lot of research dedicated to it, but has its own limitations. So it's going to need a lot of, kind of honing and perfection and optimization to make sure that we can take advantage of the gene editing capabilities. It can basically cut out a gene segment or add in a gene segment, which would be great for people who are missing a gene or who have a mutation, where we can just exchange gene segments. Wouldn't that be great? Theoretically thinking about it, it's definitely not that easy. So you have to be able to just optimize the system so that it carries out just that function and does no other deleterious harm to the other, to the rest of the genetic material or the rest of the DNA or RNA. So with that, the last thing that I want to talk about is CAR T cell therapy, and that's where CRISPR also comes into place. This is really mind-blowing, where FDA has now approved CAR T cell therapies for treatment of certain leukemias. What are CAR T cells? They're nothing but immune cells that have been taken from the body of a patient. They have been genetically modified to either add the gene of interest or delete the gene of interest, or mostly add. The gene that is added produces a CAR molecule, which is nothing but cancer antigen receptor, a cancer antigen receptor molecule. So this receptor molecule is able to go and bind to cancer cells and recognize them, and therefore be able to kill them. So you're taking patients' cells, adding a gene that can produce the CAR molecule, injecting those gene-edited cells back into the patient's body. That's when the cells produce that CAR antigen. That antigen goes and finds cancer cells and destroys it. This is I think is super mind-blowing. And now, previously, like the FDA-approved therapies used certain vectors, like bacteriophages, for trying to edit that gene of interest or insert that CAR gene of interest that would produce the CAR antigen. However, now they're using CRISPR to kind of do the gene editing here to be able to insert that CAR gene into the immune cells of the patient. So this is the peak of individualized therapy that you can think about. So, taking patient-specific cells and injecting them back kind of eliminates the immune response aspect of it as well. Whereas, if you insert some foreign cells, you will see some host rejection, and this completely solves that problem. So I think it is pretty mind-blowing as to how far we've come with cancer treatment technologies, and biomarker discovery is at the crux of it. So with that, I am going to end my talk for today. You can always find me on LinkedIn. Be sure to drop me a note so that I know, how you found me. Otherwise, I would never know. But it was a pleasure to present this to you. I hope you were able to learn something new today with my talk, and I'm happy to answer any questions that you may have with the second part or even the first part of my talk today.
Thank you, Dr. Mansi. I think this was very enlightening. Listening to some of the very cutting-edge technology work, especially the breast cancer work. It is open to questions. I encourage all the faculty members to post their questions. If you would like to, you can unmute yourself and speak directly and address it directly to Dr. Mansi. There's one question from our very own faculty, Dr. Abu Sufyan. He talks about any specific plasma biomarker of breast cancer that helps in monitoring the progress of anti-cancer treatment.
So yeah, that's a good question, Dr. Abu Sufyan. So in terms of plasma markers, That is again a very hot field, and I was about to include it in my talk, but I didn't think we'd had the time to talk about it. But what people are looking at is cell-free DNA right now. So what not only breast cancer but any kind of cancer, it's known to kind of release these fragments of DNA that are free from cells and are free floating in blood or in plasma. So, with the help of NGS and other technologies, researchers are trying to see if they can kind of identify this cell-free DNA and relate it to a specific cancer, and as well as use it as a biomarker to see at what point that cell-free DNA is being detectable in blood, and at what point we can use it as a biomarker. The challenge, though, is it's not that easy because the concentrations of cell-free DNA are usually found in very, very low amounts. So low that it is beyond the detection limit of NGS or any of the available assays. So I remember having certain projects where when I was working for a service provider company, they offered NGS services, and researchers used to send us cell-free DNA projects. The isolation of cell-free DNA is very hard. Because it's super low in quantities. It's hard to bind those DNA molecules to your isolation, whatever method that you're using, and try to elute it from the sample. Second, purity of that sample is important. Third, you need good quality of the isolated DNA or RNA to be able to run it on an NGS assay or any assay for that matter. And those isolated from these sources usually are extremely sheared and small fragments of DNA that are usually not compatible. So researchers have to really scratch their brains as to how they're going to sequence such short fragments and try to find out what fragment of the DNA is being released by the cell, what its function is, and what's happening. So it's definitely getting there. However, it is still going to need some more time and some more research for sure.
Great. Thanks, Dr. Mansi. Professor Sabha is asking in terms of some common toxicities or I guess challenges with CAR-T in cell therapy. It's a fairly new and a fairly radical way of
Sure.
treatment. But what are the common toxicities or challenges with that?
Sure. Yeah. I mean, toxicity, just thinking about the toxicity, I think overall what you're trying to do is your antigen, if it's expressed even at lower amounts on normal cells, it's going to go attack normal cells as well. That's always a risk with any cancer therapy. However, that risk is a bit lesser with CAR T cells because they very specifically go and attack antigens that are expressed by cancer cells. So in terms of toxicity, I'm really not sure specifically with the CAR T cell therapy, but with any gene therapy, there's always risk of making sure the correct fragment is edited. If you edit the wrong section of the genome, you are going to end up with off-target effects, as I was talking when I was explaining the CRISPR mechanism. So, the bacteriophage therapies and the bacteriophage method of kind of introducing that vector into the genetic material of the immune cells that have been taken from the patient is much better than using CRISPR-Cas9, and that's why I think they were able to get that FDA approval. But toxicity with CAR T therapy usually, I think, is very similar to what you would find with other cancer drugs, but a bit lesser than those because they are more targeted than just chemotherapy drugs that target. Most of the chemotherapy drugs target your DNA replicating mechanisms, and DNA replicates in all cells, not only cancer cells, but in cancer cells, they replicate faster than normal cells. So it's going to target those and other fast replicating cells of your body, like your hair, your blood, etc. So advantages and disadvantages both with the CAR T cell therapy.
Great. So as a follow-up question, a dear friend of ours, Dr. Vaishali Dixit, she's asking in terms of approximate cost. You know what? I'll ask Dr. Vaishali to kind of unmute herself, and she can have some thoughts on the this talk as well, and she can ask the question. Yes, Dr. Vaishali, so the floor is open to you.
Sure.
Thank you, Dr. Sharik and Dr. Pimple. That was an excellent talk. Some of the technologies, I should be very frank, were quite new to me too, as I'm from a traditional pharmacology field. But it definitely, opened my vistas towards what's happening on a molecular scenario. So I was just thinking, on a patient-to-patient basis, is there any approximate cost, and especially with the CAR-T therapy that you were speaking about?
Sure. Yeah, that is always the biggest concern. And along with the cost, another big concern, which I'm not sure how much it is in India, but in the US, a big concern along with cost is trying to get reimbursement for that therapy, for that treatment from insurance agencies. Because I just literally Googled the cost of CAR-T therapy, and one treatment costs about $375,000, means
Wow.
$375,000 just for one time treatment. So imagine no person, unless you're a millionaire, is going to be able to afford that kind of treatment. And if you have to, you usually rely on insurance companies here in the US to help you bail out, from that humongous medical bills. However, the newer therapies that are coming out, the biggest problem, and that's another thing that even we are facing in our company, is getting reimbursement from insurance companies. So that's a huge another process by itself. So you have to be able to generate reimbursement codes for clinicians to be able to use that easily, to get your insurance companies to pay for you. That whole thing is a different process, because it's completely untouched, and in short, it is not very easy. So you have to make sure the assay is validated. You have there's multiple considerations that go into getting reimbursement for that. So in short, that is one of the biggest limitations of these individualized or personalized medicine therapies as well. It's extremely expensive. Even getting, say for example, if patients have a specifically interesting tumor, or a tumor for which clinicians are not able to pinpoint biomarkers or subclassifications with traditional testing, their ultimate resort is to send the tumor samples for NGS, right? Next generation sequencing. Even for those, even NGS being so commonly used right now, getting reimbursement for NGS testing is also not that easy. It has to be approved. You have to justify why you need NGS treatment. The process is really hard. And imagine trying to justify why someone needs NGS to an insurance agent, right? They have no idea what that is. So all they're concerned about is why we need to pay this money, and why does your patient need this therapy when you have so many other technologies that are insurance reimbursed available. So in short, it's not easy. It's really expensive. And the hope is that with more research, we'll bring the cost down to now how we've been able to bring down costs for NGS per genome. Hopefully, we'll be able to bring down the cost for drugs per treatment as well.
Great, thanks.
Dr. Sharik, can I?
Yeah, please go ahead.
Yeah. So, thank you, Dr. Mansi, for that wonderful input, because I think it was a very frank input, especially with the concerns of insurance payments. Yes, I think we are still a long way from a common person being able to afford, but I'm sure that day is not very far away, because technology is, going leaps and bounds. It was a really wonderful session, and since I have the opportunity to speak, I would like to thank you personally for this innovative, informative session, and also Dr. Sharik, to you and your team for organizing such wonderful lectures for us.
Thank you. Dr. Vaishali Dikshit. For you, Dr. Mansi is a senior faculty at Bombay College of Pharmacy, a very well-known ranked institution in India. So we have an honor, and many of the faculty are coming from great institutions around. So we are honored to be hosting yourself and our faculty colleagues for this very interesting and lively talks that you've been going. This is the third day now. So yeah, I mean, going back to what Dr. Vaishali was talking about, this is my concern essentially. And when I used to work in US also, and almost 4 months back also, I had this, this personal, you would say, an incident or a tragedy in the family where, one of our close friend of ours was diagnosed with lung cancer, and he was given nivolumab. So the whole costing for that out of pocket in India ran into almost 30 lakhs of rupees. And unfortunately, he was a young person, and he passed away almost 15 days back. So it's a shocker for the entire family. And that just shows that the most of the new technologies and these monoclonals that are predominantly coming out as the newer therapies, are fairly restrictive in terms of the costing, especially for a country like ours, where pretty much everything is out of pocket. We don't have a whole structure of insurance for the majority of people.
Yeah,
So
yeah.
that is very concerning, where the benefits of science and the work are really not percolating to major part of the world population as such.
Absolutely, Dr. Syed. I mean, I will definitely address Dr. Vaishali's comments. Thank you for the kind words. I appreciate that. This session was helpful. Although I feel very very diminished to be talking in front of such esteemed faculty, and it's an honor for me as well. But coming back to your concern, Dr. Syed, it's an extremely valid concern. I think it's a fight between the pharmaceutical company lobbying and getting treatment at effect at reasonable cost to patients. That fight has been going on perpetually, and it's still going on, and without an end in sight. You have to kind of trade the capitalistic view of that, and then you have to trade that gets traded off with the patient centric view. But that's usually someone has to pay the cost, and in this case it's the patients who are either paying with their money from their pocket or with their lives. It's actually pretty sad, and I'm so sorry to hear about your personal tragedy. It's a whole another topic of discussion where people not able to afford therapies. And you mentioned in India we don't have the insurance structure that we have here in the US. However, it is astounding as to how much insurance companies don't help you too. And I may have a jaded perspective of that, but it is the reality. Like I don't mind sharing that I know that someone had a normal procedure that would be considered a routine outpatient surgery in the US. $13,000 was the total bill of that. Insurance decided they're just going to pay for like 70% of that, because it depends on the plan you have. Person has to come up with everything else from their pocket. And then I was told that one of the pathology services that were offered during the procedure, they let the patient know that if you go through insurance, it will be more expensive for you than if you go through out of pocket. So basically, insurance companies are not bailing us out at all, as far as I see it. But it's just the bane of the pharmaceutical industry and the field right now. I really hope that with advent of research, they will bring down the cost of such diagnostic techniques and medicines to a point where it can be afforded by people who really need it. I mean, that's isn't that the whole point? I mean, you would think people needing it is the whole point of doing research, but right now the point is for pharmaceutical companies to kind of recoup the money they spent. And mind you, they spend billions and billions of dollars too to get that drug approved and in the market and make sure they go to the patients. So they're trying to recoup that money that they kind of invested and make profit, which is why you see such insane numbers when it comes to cost for drugs. And whereas patients are like, we've been waiting for this treatment forever. We've almost exhausted all of our money on traditional chemotherapy, traditional radiation, and traditional palliative care, because a lot of them are at a place where nothing can help them. On top of that, if you tell me I have to pay $300,000 for a treatment that you think may save my life, but it may not, I don't know what to do about it. So it's a genuine concern. It's really concerning. Insurance companies are not helping make it better. So I really hope for a better world very soon. We need it more than ever.
Absolutely, absolutely. Thank you so much. Questions? Any other questions? You can post it in chat box. I know we've some thank you and thank you messages popping up, but if there is any questions, we can take it for Dr. Mansi. I can see beautiful sunlight coming on her backdrop, so it's a pretty nice Florida morning for her, essentially. If there are no other further questions, I would hand it over to Professor Saba. Before she wraps it up, I would just give you a heads up for tomorrow's session, essentially. Tomorrow we have a very interesting session lined up. I would say it's something like a mini workshop where we have 4 expert speakers coming in, essentially, and they're going to talk about a very interesting area in terms of modeling and simulation into drug development. So we'll have our very own Dr. Iyer from Bombay College of Pharmacy talks about the basics of the models as such. Then Dr. Kinjal and Amit share their expertise into the use of modeling and simulation into clinical trials. Both of them are doing exciting cutting edge work at Bristol Myers Squibb in US, followed by the end by Dr. Mike Sens. Mike Sens is also at BMS, a very senior researcher and industry expert as such, with also a 10 years experience at Bangalore BBRC facility. So he's going to have an end panel discussion. So make sure we tune in at the right time, essentially at 5 o'clock. And at this point of time, I would like to hand it over to Professor Saba for the final proceedings. Over to you, Professor Saba.
Thank you, sir. So thank you, Dr. Mansi, for very engaging session and also answering all the questions asked by our participant. Thank you, ma'am. Thank you once again. I would like to thank Dr. Vaishali Dikshit, ma'am, for your kind words, ma'am. Thank you so much. Thank you all. Feedback and attendance link is already released in the chat box. You all are requested to give the feedback of the event. See you tomorrow for the next exciting session.
Thank you. Thank you, everybody. A very good evening to all of you and a very good morning to Dr. Mansi. Thank you. Thank you, madam, once again for a
Thank you,
wonderful
ma'am.
talk.
Thanks, everybody. It was a
Thank
pleasure to present to everybody. I hope there was something new that was gained from my talk, and
Definitely.
Absolute honor. So thank you and have a great day.
We'll look forward for much more, many of these sessions going forward, hopefully.
Same here.
Thank you. Thank you. Bye-bye, ma'am. You have a nice day.
Bye-bye. You too.